Yes. So once Judy Pirog told me something like that, we do not know anything about pharmaceutical industry, about the requirements, about what kind of drug discovery tools they need or could use the best. And apparently, Judy was very convincing. So I was thinking, oh no, then how can we develop drug discovery tools for them, for our users, for you? So how actually we do that is just the typical software development procedure. We talk to the client, we try to understand the problem to be solved. We understand that to some extent. We never fully understand it. So see Judy's comment, I think he's right after all, and I agree with him. Well, as you saw, I didn't have too much choice in that, so I agree with Judy. It's very hard to understand these real-life problems, but we try, and then we create some kind of plans or design, and then we go and see our great developers. We try to make them understand what we and our clients you expect from them, and then it just happens somehow. They work hard, really hard for days and weeks, and after a while, short while or a long while, it's ready and it can be released. And that's when marketing and salespeople take over. So that's roughly the traditional software development cycle. It's a cycle because now when it's all done and is released, the user can download it and then install it and try it. And when the user tries it, that's when problems start happen, because then the user comes back and keeps complaining. Complains about the speed, the accuracy, that something else, not really what he or she expected, is not as good, is not as user-friendly, and apparently it's just full of bugs. And then an iterative procedure starts, because then, okay, we get the bug reports, we try to reproduce the bugs, and then fix the bugs, and then build a new release, and when the whole lot is put together, we can release it again, then the user just downloads it, installs it, tries it again, finds new bugs, and gets back to us, and so on in a repetition. And that's apparently very slow. So if it's slow, then we should speed it up. To accelerate the product development, we tried something in the past that worked really well, and it was something we called a sandbox. Okay, so I went to see Gabor, who was very hard at work, as usual, and I asked him to build a new sandbox, because the old one was really not used for years, and that it was also very simplistic. About the same time, we had our annual sales meeting in the head office in Budapest in January this year. Thank you, John. And during the meeting, Doug stuck a Post-it note on my laptop which said that a guy called Ignacio, who works at Takeda, needed molecular volume prediction. At that time, we did not have this kind of calculation, but we were quite confident that we could develop it. In the meantime, Gabor finished the implementation of the sandbox. It's basically a website. And he also uploaded a demo application, which was capable of generating the 3D conformer of an input structure. It could be entered either as a smile or as an SD file or pasted or a sketch by Marvin sketch. And then just you hit a button and you get the 3D structure of this input structure. So the sandbox is basically nothing else than a website. And the tools or applications that we put in can be seen without anything else than a web browser. So the user just goes there. There's nothing to download or install or maintain and update. It's all done by us. So if you go there, you always find the latest and greatest version of the product. So when this was done, I took the Post-it note that I got from Doug, and I went to see Adrian. And we had this long discussion, a kick-off meeting, and there we discussed how we could design and implement this molecular volume calculator, and we set the milestones, and we wanted to set a deadline for when the prototype can be delivered. Okay, then Adrian disappeared in the mountains. That's behind the ChemAxon office, and he started work like a hermit. Within a few days he returned. He was quite frozen, so we had to keep him warm in some blankets. But fortunately, he managed to finish the prototype of the volume calculator. Now, Gabor immediately could download it into the SANDBOX. And on the 15th of February, I informed Doug that he could contact Ignacio at Takeda, and he can send the URL of the SANDBOX, and then Ignacio can give it a try. And he did try, and he responded the next day. But unfortunately, well, as we expected, in his email he mentioned some problems related to the accuracy of the calculation. Well, I must admit that he sent other emails in which he informed us about more serious failures when the calculation just failed and it hung. But that's one example of his emails. Okay, he was so kind that he even sent us some test molecules. So we managed to reproduce the problems. We looked at the bugs and fixed all the issues that he encountered. And then we built a new version. And we didn't have to wait for the next release of the old JChem product, but we just uploaded the new calculator to the SANDBOX and sent Ignacio another mail in which we informed him that as far as we know, all the issues should be resolved. And he tried it the same day and came back to us and said that it was all excellent. And the next day, he sent us another email in which he asked for the release version because he wanted to download it and install it on his computer and on the computer of his colleagues in his group and to use it on a regular daily basis. The SANDBOX is not suitable for that. It's good for prototyping and developing and testing, but at the end, he needs the release version, download and install it. Okay, so he had to wait a few months, but on the 21st of April, he finally got the release version. So that's the time when salespeople got excited. Okay, so in a nutshell, I presented a real-life example how this SANDBOX concept was successfully used to develop a drug discovery tool that can predict the molecular volume. In this, I demonstrated that the development cycle was quite short, without the need for waiting the so-called official release. It just happened on a daily basis. I suppose, I hope that it was quite painless for Ignacio to try the newer and newer version, because all he had to do is to go to this website, paste a molecule, several molecules actually. He used about 100 in the first validation. So he just went to this drug discovery SANDBOX and looked at the results, which is quite straightforward. What's also important that by the time the tool was released, it was not only us who tested the molecular volume calculator, but the user, Ignacio, validated it for us using real molecules. It's not like when we are playing with a random subset taken from the NCI database. He really used his, those molecules that he was working with. And at the end, when the release was built and he downloaded it, he got really what he wanted, exactly that piece of software. Now, all this also ensures that in the release version, we can expect much fewer bugs, not only reported by Ignacio, but others who pick up this tool and use it. And soon after, the SANDBOX was successfully used to develop another calculator that's called the minimal projection area, which has been applied to predict blood-brain barrier penetration by the collaborating customer who asked us to develop this calculator for him. That's also been released since then. And actually, you could see that in one of Alex's slides during his demo of the chemicalize.org. Now there again, we use the SANDBOX for rapid prototyping and for a responsive development to shorten the product delivery cycle. Okay, now let me overview what else has the drug discovery tool team developed in the past 12 months, so let's say since the last US UGM. And actually, these all that I'm going to show you now will be released in the next major release 5.4. A year ago in San Diego at the second US UGM, we've been told that the industry standard molecular force field for various calculations like molecular mechanics, dynamics, and energy minimization, energy calculation was the Merck molecular force field. So that's Pat actually, and Pat told us that we should forget about the ChemAxon force field that we were heavily developing in those days, and we should implement just the golden standard, the Merck molecular force field. Okay, so when we returned back home, I went to see Ödön and Laci, two other hard workers at ChemAxon, and I managed to force them to implement the Merck molecular force field. And it's done, and it's been validated against the trusted MMFF set, which is available for download at the Cambridge Crystallographic Data Center. And in these validation studies, the accuracy of our implementation was below 0.002 angstroms in RMSD. And the speed is comparable to the speed of the ChemAxon force field. On average, it takes 3 milliseconds to minimize the energy of a structure over the MMFF force field. Also in San Diego, Oleg presented his study in which he compared various fingerprints against each other, including the ChemAxon fingerprint. The ChemAxon hashed chemical fingerprint. While he found that, for instance, the cytogic fingerprint performed really well, our produced 0 enrichment in his tests. Well, at that time there in San Diego, I didn't comment on that, but now I really have to. So it's because this ChemAxon hashed chemical fingerprint was developed to speed up the substructure searching. So this fingerprinting technology is an integral part of the JChem search engine to do pre-screening, and thus it's less suitable for similarity calculations, for which Oleg used it for. Where are you? I can't see you. Anyway, we returned back home, and I grabbed Peter, a talented young developer at ChemAxon, and gave him all the publications about the circular fingerprints and asked him to implement the ECFP, the Extended Connectivity Fingerprint, or its popular name is the CyTG or the pilot fingerprint. Now, this work has been accomplished since then, and both ECFP and its enhanced version, the FCFP, will go into the next major release, 5.4. Oleg also said in his talk that he implemented the maximum overlapping search algorithm. At that time, we only had something we called the maximum common substructure search, which is something similar but simple than the maximum overlapping set, or MOS. Although our implementation was quite fast, but from the feature point of view, MOS is a lot better than MCS. So this made me very envy. And though I had concerns regarding the performance of an MOS search, yet we decided that we should implement MOS. And Oleg was a great partner to collaborate with, because he sent his program for us, so we could try it. We could see how well the MOS is identified by his algorithm. And he also sent us the publication, which he implemented basically. So the publication outlined a search algorithm for this MOS, and that's what Oleg implemented, and he sent us this paper. So again, I went to see Peter, and I gave him this paper that we got from Oleg, and he implemented a similar MOS algorithm. This will soon go up into the sandbox, and if all the validations are successful, then it will be released in 5.4. In 5.4, we will deliver shape similarity-based 3D screening. This is something like ROCS by OpenEye. Now, you need to know that ROCS really set the golden standard in ligand-based 3D screening, so it's very hard to compete with it. Running times, which are important in this application field, are about 10 times better than that of ROCS. And regarding the accuracy, this chart shows the enrichment ratios compared to other products, among them, most importantly, ROCS in blue and ChemAxon in orange. Bear in mind that these are really the first results. The evaluation of this 3D screening and the fine-tuning of the program is still in progress. But you can see that even these first results are encouraging. This 3D screening technology performs as good as the competing products. So 3D screening will probably be released in 5.4, and the 3D screening will also be available in the drug discovery tools sandbox. It's Gabor who keeps putting newer and newer tools in the drug discovery tools sandbox. Recently, he implemented the new version of Library MCS, our popular structure-based hierarchical clustering tool. But now he incorporated the MOS search, which gives great advantages over the MCS search. So perhaps now on we need to call this tool Library MOS. I don't know. But there's room for many more in the sandbox. We have ideas like more shape descriptors, advanced virtual screening, new multi-conformer search based on a new molecular dynamics approach that we presented in our 3D poster. We are thinking of a new user interface for clustering, new cluster visualization techniques, the BBB penetration, other membrane penetration predictions, and so on. But what really goes into the sandbox depends on you all. And most importantly, what comes out from the sandbox is also influenced by you. because we built this sandbox for you. So come and play in it. And please, please collaborate with us in developing useful drug discovery tools. Thank you.
For the rocks example, there you need to probably deconvolute the omega part. So were you using your conformation generator or theirs? And you probably need to deconvolute those two and try each with each.
Yeah, we don't need conformal generation, unlike rocks. I mean, we do not need to generate a given number of rigid conformers, like 400 is a typical with rocks. But instead, we use a continuous, fully flexible conformational search, conformation analysis during the screening. So our starting conformation is just one single 3D conformation, whatever that we generate with our conformer generator too.
Did you publish that already?
No, not yet. Yes, it's just discoverygroup.chemaxon.com.